We are the Group 5, and this is our final output in biochemistry.
Relationship between lipogenesis and citric acid cycle intermediates. The citric acid cycle, or we can simply say the Krebs cycle, serves as the mitochondrial hub for the final steps in carbon skeleton oxidative catabolism for carbohydrates, amino acids, and fatty acids. Each oxidative step, in turn, produces a coenzyme such as nicotinamide adenine dinucleotide or flavin adenine dinucleotide. These reduced coenzymes contribute directly to the electron transport chain and thus to the majority of ATP production in the human body. The citric acid cycle is a metabolic pathway that connects carbohydrate, fat, and protein metabolism. The reactions of the cycle are carried out by 8 enzymes that completely oxidize acetate in the form of acetyl-CoA into 2 molecules, each of carbon dioxide and water. Lipogenesis is defined as the synthesis of fatty acids from non-lipid precursors. It is a pathway for metabolism of excess carbohydrate and is activated by high carbohydrate availability. The creation of fat, more precisely, the process by which acetyl coenzyme A is converted into fatty acids in living organisms, particularly in the liver and adipose tissue.
The relationship of the two. The citric acid intermediates involve C4 diacids, and the lipogenesis intermediates involve C4 monoacids. The order in which the various acid derivative types are encountered in lipogenesis is the reverse of the order in which they are encountered in the citric acid cycle.
Fate of fatty acid-generated acetyl-CoA. Cells normally have to change during famine from growth to survival mode and modify metabolism to perform tasks necessary for survival. Under these circumstances, nucleocytosolic acetyl-CoA levels fall because there is a larger need for acetyl-CoA to be oxidized in the mitochondria for ATP generation rather than exporting acetyl units to the cytosol. An important source of this mitochondrial acetyl-CoA pool is fatty acids. Fatty acids are activated by fatty acyl-CoAs, which are subsequently carried into mitochondria by the carnitine shuttle for B-oxidation. This process is known as CoA production. As a result, the mitochondria produce acetyl-CoA, which may be oxidized or have other uses. Acetyl-CoA in the liver's mitochondria produces ketone molecules, such as acetoacetate and B-hydroxybutyrate, which serves as substitute energy sources for the brain and heart under the conditions of carbohydrate scarcity.
Relationship between lipids and carbohydrates metabolism. Fats and lipids are essential components for the homeostatic function of the human body. Lipids contribute to some of the body's most vital processes. Lipids are an essential component of the cell membrane. Lipids and carbohydrates are 2 of the most important biomolecules in our body. While lipids are essential for energy storage, cell membrane structure, signaling pathways, carbohydrates are the primary source of energy for our body. The relationship between these 2 biomolecules is complex and important for maintaining a healthy metabolism.
The interaction between lipid and carbohydrate metabolism is complex and depends on various factors, including the type of quantity of food consumed as well as the level of physical activity. For example, consuming a high-fat diet can lead to an increase in triglyceride levels in the bloodstream, which can impair glucose uptake by the cells and lead to insulin resistance. Similarly, consuming a high carbohydrates in blood glucose levels, which can promote the synthesis of triglycerides and lead to an increase in storage fat.
So we come now to B vitamins and lipid metabolism. Vitamins play an essential role in lipid metabolism reactions, and their presence is therefore absolutely necessary for this reaction to occur. The effect of pantothenic acid, niacin, and riboflavin is here described. By transformation into coenzymes, these vitamins are involved in fatty acid synthesis and oxidation reactions. Other vitamins like vitamin B12, folic acid, vitamin C, and essential fatty acids influence lipid metabolism by different mechanisms. Coenzyme B12 and folate coenzyme provide the balance by methionine synthesis and the pool of methyl radicals necessary for phospholipid biosynthesis. By its involvement in the microsomal respiratory chain, vitamin C promotes cholesterol transformation into bile acids. The essential fatty acids, mainly linoleic acid, are directly connected with cholesterol transport and plasma cholesterol decrease. Vitamin B12 is a cofactor for the mitochondrial enzyme methylmalonyl CoA mutase and regulates the rate of long-chain fatty acetyl CoA transfer into the mitochondria, leading to the accumulation of fatty acids in the cytosol, thereby influencing lipid metabolic pathways.